I just put this picture here to recall your memories on Darwinian evolution. This picture represents the last 4 million years of human evolution, and this time was needed to evolve from Australopithecus afarensis to the modern human Homo sapiens. And this Darwinian evolution process takes place in the germ cells, and one of the speed defining factor here is the rate of gaining new mutations in the germ cells from generation to generation. And a growing body of evidence shows that in a single human body we have extensive variation, genetic variations among somatic cells. So one human has in fact numerous genomes. And this is the basis of another significantly speeded evolutionary process within a human body, which takes place in somatic cells, and this is the tumor evolution. Tumor evolution is driven by the stepwise accumulation of so-called driver mutations. These driver mutations conferring growth advantage to the tumor cells, cancer cells in our body. Practically here the generation time is much shorter because one cancer cell can divide even once a day. And here also the speed defining factor is the rate of gaining new mutations from cell generation to cell generation. Definitely this can be much higher as compared to the Darwinian evolution because we don't need to evolve a complete functional human body from a cancer cell. So there is a special group of driver mutations which are directly influencing the speed of gaining new mutations during cell divisions. These are the so-called mutator mutations, and they occur in the so-called caretaker genes. Caretaker genes are genomic guardians. They are guarding the integrity of the genome. So the mutations in those genes are able to increase, so they're able to increase the rate of gaining new mutations. So our laboratory is particularly interested in studying such kind of driver mutations. We study this type of driver mutations and the carcinogenesis itself in vivo model systems, and we model this type of mutations by introducing transgenes into our model system, and to modelize oncogenic mutations, we overexpress mutant cDNAs, and for tumor suppressor type mutations, we express artificial microRNAs silencing our candidate tumor suppressors of interest. And also we are using random insertional mutagenesis, and all of these transgenes are introduced with the DNA transposon systems, in this case with the Sleeping Beauty transposon system. The Sleeping Beauty transposase helper protein is administered in trans, and it binds to the small inverted repeat structure surrounding our transgenes and cuts out this transgene from the plasmid DNA environment and integrates into the genome of the host cells to almost in a random manner to any TA dinucleotide. So as I mentioned, we are studying this type of mutations in an in vivo system. I just put here for comparison the conventional germline transgenesis, the two most frequently used applied protocols for germline transgenesis, the ESL-based and the pronuclear injection-based protocols. Without going into the details, these are very time-consuming, and one needs a big animal host to be able to practice successfully these techniques. So in contrast, we are using a somatic transgenesis method. This is in fact a gene therapeutic approach. It's a one-step transgenesis in one organ, and this is much quicker and better reflects the somatic occurrence of driver mutations. So on the basis of this protocol in our laboratory, we have two branches of efforts. One is to use this kind of protocol to study carcinogenesis itself, and the other is to develop further this gene therapeutic protocol to better fulfill the needs of modern gene therapy. Our first year Szegedi student Réka Karkas is involved in improving further this gene therapeutic method, and she's an extremely talented student, and I'm sure she will be able to fulfill all the criteria in the future to be remaining a Szegedi fellow. We have two international lines of collaborations, one with Professor Thomas Rülicke in Vienna, and another with Professor David Largespada in the USA. David Largespada is an American Cancer Research Society research professor, actually. So now please listen the lecture of our Szegedi student.
Dear audience, I would like to talk about the in vivo gene therapy in the mouse model of tyrosinemia type 1. Tyrosinemia type 1 is a fatal disease with progressive liver dysfunction. It is caused by the germline point mutation in the fumaryl acetoacetate hydrolase gene. The product of this gene is the last protein in the tyrosine catabolic pathway. The lack of this enzyme causes accumulation of mallyl acetoacetate and fumaryl acetoacetate, which are converted to a highly hepatotoxic metabolite called succinyl acetone. There is a drug called NTBC, which is a pharmacological inhibitor of the tyrosine degradation pathway. It inhibits the hydroxyphenylpyruvate deoxygenase. In our model of this disease, we keep our FAH deficient mouse stain in this drug till they receive treatment. For example, our in vivo cell replacement system, in which we isolate hepatocytes from a healthy male donor and inject them into an FAH deficient female recipient. After we remove the NTBC, the donor-derived cells gain selective growth advantage, so they are able to repopulate the liver, so the liver can show extensive regeneration process. So after 10 weeks, almost 95% of the hepatocytes are donor-derived. We can identify the Y chromosomes in these cells by fluorescent in situ hybridization. The weight curve shows that the placebo-treated animals without the NTBC die within 5 weeks after continuous weight loss, while the treated animals, after an early period of losing weight, start to show that liver regeneration process, and their weight is progressively growing. We utilize this approach to create an ex vivo gene therapy system in which we isolate hepatocytes from an FAH deficient donor and electroporate them with a Sleeping Beauty transposon construct, which contains a fluorescent marker gene and the therapeutic FAH gene linked with the T2A peptide, which enables ribosome skipping through the translation of this two separate protein. We also add Sleeping Beauty hyperactive version transposase to the system. And after that, we transplant these genetically modified cells into an FAH deficient animal. After the removal of the NTBC, the corrected cells show that repopulation process, and they co-express the fluorescent M-cherry marker and the therapeutic gene. Beside this randomly integrating transposon-mediated system, our plan is to create an effective site-specific system so that we utilize the RNA-guided DNA and the nuclease system. This CRISPR-Cas9 system contains a Cas9 nuclease which forms a complex with CRISPR RNA and a transactivating CRISPR RNA duplex. This complex is able to bind to a specific 20 base pair long target region and create a double-strand break in it. I have learned the basics of molecular cloning and using bioinformatical softwares through creating and planning these gRNA and Cas9 expressing vectors. The Cas9-induced double-strand break can be repaired by homology-directed repair. This pathway requires a repair template, which is copied into the cut target sequence after the homologous recombination. So in our donor template plasmid contains a sequence to be introduced between two homologous arms. After the repair process, we gain a functional FAH gene. Our plan is to optimize this system to decrease the off-target effects of the system and increase the homology-directed repair pathways dominance in order to create an effective and site-specific gene therapy system. I would like to acknowledge my mentor and my supervisor and all the staff members in the Laboratory of Cancer Genome Research for their generous help. And I would like to thank you for your attention.